So is my screen visible?
Clear, sir. Clear.
Yeah. So hi everyone, I am Deshank Purandare, and today I am just going to give a brief knowledge about what molecular docking is, why is it necessary, and why is this topic, taking a huge jump and becoming a hot topic in India. What are the regulatory changes that have happened in the last 2 to 3 years, due to which molecular docking is now being, the most essential part for all the pharma companies and all the biotech companies. So, just to begin with, this is the summary of today's session. So, initially, I will be introducing you with what exactly molecular docking is, what are the different types of molecular docking, how to identify your protein and how to prepare it, then how to identify and prepare a ligand. The most important step that I will be focusing on are these two, is the identification and preparation of a protein and identification and preparation of a ligand. And then how we can see and how we can visualize these interactions and the regulatory guidelines. These are some of the proteins here. This is a protein, this is a ligand, and this is the visualization that we will be seeing later on. So, what is molecular docking exactly? It is a computational process wherein a ligand it fits onto the active sites of a protein. Like we all know, our body has a lot of proteins made up of amino acids. These amino acids and the protein have some active sites on them. In order to a drug, suppose there is a drug that we usually call as an active. This suppose a paracetamol in a general example to in order to make you guys understand what exactly I'm talking about. We usually take paracetamol, medicine in order to reduce our fever and in some cases as an analgesic as well. So how exactly does this molecule interact in our body is that it goes and binds to the active side of the protein and then it gives its action. And then the antipyretic or analgesic effect that's been required. So over here, let me show you a phase wherein how a drug is being developed. It usually takes around 14 to 15 years just to develop a drug, and this process is very time consuming and requires a lot of finance that goes into this. There's you can see the discovery phase, it lasts around 3 to 4 years, then it goes to the preclinical trials, then regulatory filings, then clinical trial. Over there also phase 1, phase 2, phase 3, and then the finally comes the regulatory approval and the drug comes into the market. So this usually it takes around 14 to 15 years. But how can we reduce this time? Because as the time goes on increasing, the finance that goes into this, the cost of developing the drug also keeps on increasing. There's when the picture of molecular docking means the molecular docking comes into the picture. So how does it help to reduce the time? Is that over here, before molecular docking, what used to happen is that a chemist used to synthesize a drug. He used to test it, results come, okay. Results are not there as per the requirement. Again, he used to go back, synthesize some other drug, and then test. Over here in molecular docking, what happens is that before synthesizing, we can test nearly about thousands to lakhs of compounds and then only select those compounds for synthesis and then animal studies and then human studies which show a potency. This is how we can identify the lead compounds and streamline the drug development and drug discovery process. So what are the types of docking? Usually there are more than 5 to 10 types of docking. Today I will be touching only 2 to 3 types which are widely used in the industry. So first one comes is the flexible docking. Over here in the flexible docking, the ligand. or the drug, it adapts to the position of the protein. Like suppose a protein may have this, you can, if you can see, this is a protein. Suppose there are five active sites. We don't know what exactly is the active site or how to identify it. That's where the flexible protein comes into the picture, wherein the ligand gets attached to all the active sites, and then we can select a favorable position and the favorable orientation. Then comes the blind docking. Over here, I will show you further ahead what exactly I'm talking about. Just to give you an idea, a blind docking is where the entire surface of the protein is being treated as an active site, and it helps to identify unknown pockets and predict the active sites which might have a higher potential compared to the active sites that are known to us. Then this is the type of docking that is been widely used, induced split docking. Over here, not the entire surface of the protein is not taken into consideration, but only it is used to identify the unknown pockets and also to verify the active sites that are been already known to us. So now let us start how to identify a protein. So initially, you need to know some biological parameters like what the target is or what is the disease pathway. Suppose you are working on cancer, let's say lung cancer. There are more than 5 to 10 pathways that you can target, such as thymidylate synthase pathway or EGFR pathways, VGFR pathways, etc. So initially, you need to know what exactly is the active site that we are talking about, or what is the pathway that we are going to target. Then we come to the literature review. So then the 3D protein, the database, the PDB database, we retrieve a protein there and then select the active sites and the actual process begins. Now I will show you a simple uh, yes. Now we will go to the PDB site. The first step over here, the identification of the target, the literature review, and retrieval of the 3D structure from the PDB database. So this, for your knowledge, is a PDB database, how it looks. And from here, we can search different proteins of our choice. This is something that the site looks like. I will show you what exactly I'm talking about so that, it will be a bit interesting for you. So initially we come to the PDB database. This is the PDB database. Over here you can see a lot of proteins going on. A lot of scientists all around the world, whenever they discover a new protein, a new active site, they usually deposit their proteins over here. So you can see there is a deposit site as well. So now 6ZXO. Now what I'm typing over here, what is 6ZXO? So 6ZXO is a protein that I have identified, and it works in the thymidylate synthase pathway in order to cure the lung cancer, most precisely the non-small cell lung cancer. It is usually, found as a very active and very effective site, the protein to act on. So from here, we download the PDB format of our protein, this PDB file we do download. I will show you what we do with this file later on. Just let me finish the theoretical part first. So now we come towards the ligand. What exactly is a ligand? Ligand is nothing but the active protein that we are talking about. So how it is been done? Initially we take the ligand usually from a pubchem if the ligand is well known. But if you make some novel ligand or not novel active compound, which is very novel to you and it is not been present anywhere else, then we use the ChemDraw and Marvin sketch that I have written over here. We then convert the 3D structure in order to generate a stereochemical structure or a stereochemistry of that molecule. We then minimize the energy in order to make the ligand stable and to obtain a more stable conformation. Then we reduce the partial charges, we remove the water molecules, and then we convert that ligand into a PDB file. This is the PubChem that I was talking about. Over here, you can search any ligand of your choice. Let me show you. Here, suppose you need a metformin structure. Metformin is a well-known compound used in the treatment of diabetes. So from here, you can download the SDF file, this, and which we later on use in order in the docking process. I will show you exactly what I'm talking. This the 3D conformer that I was talking about. Then we come towards the ligand preparation. Now, like I said, this SDF file, when we save, we cannot directly use this file in the molecular docking process. There are a huge number of steps that need to be done before we take the molecule for docking. Firstly, we use a software known as a PyMol, wherein we introduce our structure in it. Then we export the structure into the PDB file. Now, let me open up the PyMol software for you so that you get a better knowledge about what I'm talking about. Yeah. So this is the PyMol software's interactive phase or the phase that you see. Yeah. So the SDF file that we have downloaded earlier. Let me show you how it's being done. The SDF files that we had downloaded from the PubChem, we usually drag and drop it into the PyMol. Here, you can see the structure that we had downloaded. 3D structure of the ligand is now there on the PyMol. From here, we export this structure to the PDB file, the exact same file that we use for our protein, and then we save it. Now you have your protein, you have your ligand, but we also need to stabilize both our protein and ligand as well. So what we do is then we move on to the molecular docking, the actual molecular docking using the Autodock tools. Over here on the left side, you can see the ligand that we had, you know, converted the file from the SDF to the PDB format. We have uploaded it over here in the Autodock tools, and then we, usually from the grid, you can see over here. We usually, if it is a flexible type of docking that we want, then we convert the non-rotatable bonds into the rotatable bonds. And then if we want a rigid docking, then we convert the rotatable bonds into the non-rotatable. And over here, we can, stabilize the protein. So now you have your protein, you have your ligand. What next? Then usually we need to write this code using the CMD. It may look as a very difficult thing, but it is not. Just the process is process that goes into it are lengthy. And once you write this code, you do get something like this. Now, what exactly is this? Is there over here? You can see the docking score of the various. This is the single molecule. Over here, since the active site was not known to us, we took the 9 different positions that were recorded. And from here, the higher the number in negative, suppose this 9 we usually take this and then we move ahead for the visualization. For visualization, we use Discovery BioVia Studio. Over here, when we do the docking, we get this output file. We do upload it over here. This is some view that we get. All these things that you see are the individual ligand, and you do see a cluster of them because of the protein, the active sites. This is the protein, and then we see the interaction of both the molecules. Once this is done, you can check the amino acid interaction of both of them. And now let us move towards the practical part. Yeah. So over here you can analyze a lot of different things. You can also do MD simulation. I will tell you what those things are. Uh, our concern thing is over here, the receptor ligand interaction. You can also see different pharmacophores. Let me just upload this. Yeah. So this was the protein, the 6ZXO that we had initially downloaded from the PDB database. I will get a very clear idea what were the things that I was talking about. So over here you can see A, B, C, D, E, and F. These are nothing but the chains of the protein. Suppose if I want to remove the F, so I will just click over here. You can see that the F chain of the protein is been deleted. This usually you need to research well and do your literature survey well before removing or adding any chain to your protein. Over here, when we specifically talk about this protein of 6ZXO, all the 6 chains that you see over here, A, B, C, D, E, and F, were the active chains, and the active sites were present in all of this. As a result, I have kept each and every chain over here. Then we take our output file that we obtained after performing the molecular document. See, You can see that where our active site or was and where our ligand is now being interacted upon. So from this, what can study further is that you can go to the ligand interactions. These are some of the 3D interactions that you see. Uh, this is the aromatic centers that you can see. Usually, all the things that I'm talking about might go above your head, or might, you might feel like you are not getting the points that I'm been saying. But whatever is going on over here, it usually it is a step in the drug development process. And, now if we see the hydrophobic sites, from this, you can develop a drug. Suppose you need to target disease like Alzheimer's, or you need to have a blood-brain barrier crossing ability of the drug. So from these interactions, you can see where the hydrophobic sites are, where is the hydrophilic region, and then accordingly, you can develop your active sites of your drug. Now this happened. This was when you have a molecule that is already available. But what if the molecule is very novel to you? Then how do you go about? And for that we use ChemDraw 3D Pro. Over here, uh, a novel drug, suppose you have developed, whose structure is not available on the any sites, such as PubChem and all. So what you can do is using the ChemDraw 3D, you can develop the or draw the structure of your molecule over here, and then convert it into the SDF files. Import file, if you go, then you can develop your own molecule and then follow the similar process of the drug development. So these were the acid interactions that we saw over here. This, if you see, if I just remove this item, see this distance has been written. So all these are the amino acids that are been interacting with our drug molecule. This thing that you can see are the amino acids, and the one marked in the yellow color is our drug. And this, it is very interesting thing to see this electrosteric bond, hydrophobic bond, the hydrogen bond. They are residues, amino acids, which were interacting. And this is the binding energy that I initially showed you. So why is it necessary right now? So in the US FDA, from the corona, after the period of COVID, they have been mandated this that the in silico studies are being mandated. EMA, the European Medical Agency, who regulates the European market, they have also made it compulsory for molecular docking studies to be there. Then in Japan also from 2019 they have they do encourage the users and manufacturers of the pharmaceutical compounds to do the molecular docking. And then the India, it is not strictly being followed, but it will be. It is just a matter of time. What happens when now you know what exactly the things are going to be. So what are the job opportunities for you? So, computational chemist, a simple person who can do a docking can be paid around 10 lakh rupees per annum. This is usually the salary ranges once you have 2 to 3 years of experience. And companies like Pfizer, Novartis, they take you uh, in order to perform the QSAR, the NP simulations and all. And molecular docking scientist also Schrodinger, Autodoc Vina that I showed you. They pay you around 16 to 18 lakh rupees per year. if you want to, learn the things, because now it was a very quick session. But if you want to learn exactly in detail what are means how to prepare a ligand, how to prepare a protein, and how to perform molecular docking. So there is a 7-day international workshop which has been starting from the 6th December onwards. So you can register over there. Over here, along with me, experts such as Savish Polshettiwar sir and Shrikant sir. These two are very expert in the field of molecular docking. So along with me, these two advanced professors will be there. Along with this, Mr. Sujit will also be there. So you can register over here. And here are some of the publications that uh, I did in my college level. So over here, the 6-ZXO protein that I had showed earlier. So it was used in order to study the non-small cell lung cancer targets of various drugs. Then again, the non-small cell lung cancer over here, we use the P13K protein. And this was again a triple negative breast cancer, wherein a lot of people were involved and good results were shown. And then this was again for the breast cancer EGFR inhibitors. Now let's have some questions. I know the session was very quick, but if you have any questions, we can take them now.
Okay, any questions with respect to molecular docking? Can ask with respect to questions related to career options, your any queries related to your product you are working on, anything.
The session was a bit, quick, but anything that you need to ask,
I guess very many of them are from some of the students who or candidates who are joined are from bioinformatics field also. So they are aware of this molecular docking,
Yeah,
the phenomena, the importance of molecular docking.
yeah.
So any questions? You can ask in the chat box also. Anyone? Any questions? Priya, thank you so much. Hope the session was useful to you. Dishan, can you see the questions in the cell in the chat box? There are two questions now. You are on mute, I guess.
Yeah, now am I audible?
Yeah, please.
Yeah. So Parag has asked what are the types of docking. So usually there are, more than 10 to 15 types of docking. But the thing that we really use in the industry are the three types that I told you. First is the flexible docking, wherein you have no idea where the active side was. Like I showed you over here in the 6ZXO. This. So there were six chains that I showed you initially. So over there, I knew where the active side was. As a result, I did not go towards the flexible docking. Rather, I took the rigid docking approach. But if you don't have a clear idea of where the ligand shows an optimum binding and can give us the good results, Then we go towards the flexible docking. This blind docking is when you have no idea on you that the protein has any active sites or not, and you need to find all the possible sites and all the possible things. Means suppose there will be n number of binding sites on that protein. Then we go towards the blind docking. And this induced field docking is also known as a rigid docking, wherein you know where your active site is. As a result, convert all the rotatable bonds in your ligand in a non-rotatable bond, and then you perform the docking studies. These are the three major type of docking that we usually prefer and usually perform. And then one question. Let's go to the molecular docking. Have to teach Yeah. So usually for herbal extract, what happens is that the molecules or the ligand are usually so bulky that to dock that molecule towards a specific protein is a big challenge. And if you can do it, it is a very novelty that you can claim, and you can always also file a patent on it, because if you also means if you search on the Google Scholar or any research paper, you will find a very few papers where perform docking on a herbal extract. The thing first is that your herbal extract, you don't exactly know to which active site it is going to bind, which mechanism or which pathway it is going to follow. As a result, selecting a protein for your herbal extract is a challenge. Second, your herbal extract, if it is not a bulky group, suppose its molecular weight is less than, say, 900 or 1000 daltons, it is fine. But usually, the herbal extract, their molecular weight is above 1000 daltons. As a result, it becomes a challenge, but it is not like you cannot do it. We can always do it. What we can do is that we can break the ligand into small, small fragments, and then there is a thing known as a fragment docking, wherein small, small fragments are being taken together, they are docked, and then in the end, you take everyone together and then you dock. So you can do it. And saying about the limit on the ligand. So usually we take around 10 ligands we do take, but there is no as such as a written limit on how much ligands can you take, how much documents the ligands can you doctor for analysis. Mainly is possible. So for analysis, the last step, which is the visualization, which we did using the BioVia. That's the main, the most important step is because, like I said, see over here, there are ligand interaction. You can also study the pharmacophores, X-ray studies. So once you have your output model with you, which is this, you can do n number of studies with it. It is not just specific to getting a docking score or seeing the amino acid interactions. You can do n number of studies with this. So the final step, which is the visualization thing, is the most important step. What are the challenges that the major challenge that we face during molecular docking is getting the right protein for your ligand. You may prepare an excellent ligand, which may have an excellent means activity when tested on humans. But if you don't know which exact protein or which exact site is optimum for your ligand, then you may not get the optimum results, even though your molecule may be highly potent. So the first challenge is selecting the right protein. The second is choosing between whether you want a flexible type of docking or a rigid type of docking. You may have a protein, for which you may know that my protein has two active sites. But and then if you go for a rigid type of docking, so things can happen that it may bind to some, second, means out of the two, it may bind to one. But it is always beneficial that you go for a flexible type of docking wherein you can or perform a blind docking. The use means the advantage of performing a blind docking is your ligand will go to each and every corner of the protein and then select the best and the most optimum site in order to perform to give you a better result. These are the two main challenges that we face. First is the selecting the right protein, and second is the selecting the right active site.
Shashank, one question was left out from the first one. Anuja had asked.
Can you give an example of molecular docking monoclonal antibodies? So, in monoclonal antibodies, we just, there is only one change that we perform, which is instead of ligand, we take your active, vaccine or the active part of the dormant virus or the antibody. And then the further thing that we do is stabilization of the protein, finding the active site. It all remains the same. You just need to have the mono and let me just show you over here. If you go to macromolecules, you can using the Discovery BioVia Studio. So you can build your own nucleotide. You can build your own this monoclonal means the antibody for a simpler example. And uh, this ligand that we have initially taken up from the PubChem. So instead of that, you can build your own antibody over here and then clone it, visualize it with the protein. And then this is how you can get your optimum results. And be sure that whatever you are doing is actually correct. How do you choose binding sites of protein? Multiple. So, if the protein has a multiple pockets, so if you are performing a rigid docking, then the question won't be there of the different sides, binding sides. But if you are performing a blind docking, then over here you can see when we write this code, we also we do perform a config file before writing this code. So in this config file, I will show you. See, the energy range and exhaustiveness. Usually we set it to 8 such that it can go and move around the whole protein and then select the active site. The question that if a protein has a multiple site, how we select the optimum or the best site is based on the docking score. This is based on the docking score. The first mode that you see uh, shows us a binding energy of minus 9.9. Then it goes to 9.4, 9.4, and this. So 9.9 side was the highest or the side that showed us the highest or the optimum energy. Once we bring this output model in our Discovery BioVia Studio, so the first model that had shown, see, you can see that a molecule has been highlighted in the yellow itself. This was the molecule that binded to the active site of your protein. And once you take this molecule over here to the protein, then you can see where the drug binded and showed the optimum activity. You can see one drug had actively binded over here, some of them had binded over here, but the optimum energy was showed by this molecule. We then specifically select this molecule and then we transfer it to our protein and then we get something. So that's how you can select the active site. Then we have a multiple pockets. And, I can tell that the molecular document blind document. So we usually go with the blind document itself, whether you know what the active site is or whether you don't know where the active site is. But at a college level or at a master's or a bachelor's level, you don't need to perform those extensive studies. As a result, we go with the fixed document. But generally in the industry, irrespective whether you know what the binding side is or you don't know what the binding side is, we go and we perform with a blind documentation. So, all the things that I, showed you over here in my PPT was a very quick go through to the different things that we do. We want to actually get to know how it's done stepwise, getting access of all the softwares. The thing that I showed you from the 6th December, a session of 7 days is being, taken, wherein with me, two other experts are there along with Sujit sir. And then over there you can, get to know every small point. What exactly is a blind docking? What is a, flexible docking? How can we select the active side, the best optimum side? If the protein has a different multiple pockets, how to select it? So over there you will get to know in detail what exactly are the things that I am talking about and how the industry works when it comes to molecular document and this. Another question or anything that I have missed?
Okay, I guess all the questions has been answered, right?
Yeah, yeah.
So thank you so much, Dishang, for taking time out on this weekend for this session. And as
Yes.
I guess almost 28, 30, 33 candidates had already joined for this session, and they had stayed throughout the course of the session. So we can be sure that they are the people who really look out to or understand the importance of molecular docking.
Yeah,
So as Dishang has already mentioned, we will be coming up with a 7-day workshop. It's an extensive workshop which has been arranged based on inputs received from candidates, and many professors had requested to arrange this session. So we had arranged it, and also the fees had already been kept nominal compared to what almost we are giving at least I guess 4 to 5 software access in during the session. We will be conducting it as an internship also, means so that you will be taking hands-on experience with respect to the topics. That's why we want you guys to attend it. And if you are facing any issues with respect to the fees part, means we have already given discount of around 50% already has been announced with respect to the fees part. And if you still feel that with respect to if you are facing any issues with respect to the fees, that shouldn't be a barrier in your learning process. So you can contact me or anyone in the team. We can make suitable arrangements. But take advantage of this session because we want you to take hands-on training on I guess if I'm not wrong, Dishank, please correct me if I'm wrong. We are having arrangement for 4 or 5 softwares, right?
correct. All
Yes,
the softwares that I talked today,
yes,
so
yes.
we will be giving them the access of all.
Yes, so you will be having access, and we will be working out on a means after your sessions in colleges or offices are over. That's why we have kept a time of 7:30 to 9, and I don't think any other particular requirement is there from their part. They only require a computer or a mobile also will do for
A,
accessing it.
yeah, yeah, a basic laptop or a basic computer is more than sufficient.
Yes, yes. So you will be having access before you enter the industry. You will be having access to the softwares which are used actually by the industry. Trust me, it would be it's going to be a game changer session. That's why we have kept 2 speakers exclusively for this, and 2 speakers from pharma, means including me. so that we will be providing inputs with respect to the pharma industry and also the application part. So that's how the arrangement has been done. We have two software experts and two speakers who can address the industry from an industry part of it. Okay, so I'll thank each one of you who have attended this session on a weekend. It's not easy to attend sessions on weekends, but you have all attended it and are making this series a very or making it very successful. I will be uploading this this particular video on YouTube also. You can, as part of you, if you want to revise those concepts which Dishang had already explained, if you want to go through the topics again, you can go through, you can hear that topics in YouTube again. Okay, thank you each one of you. Special thanks to Dishang. We'll conclude the session here. Thank you. Okay.
Thank
Good
you.
evening, everyone. Bye.